is feeling like I should really lecture recombinant DNA in person and leave proteins up to you.
Probably.
I think that's my feeling because of where we're at. So I'm going to leave proteins for you to kind of go through because I think it's more straightforward. Like you can look at the slides, read it, and there's some videos that can help you, and I think it's okay. But I think recombinant DNA, I would feel better if I just walked you through it and asked you some, made sure that you asked questions and understood what was happening, okay? So make sure you go back and finish up that proteins lecture, proteins and enzymes, review. And I will update the study guide with what I really need you to focus on for that lecture. But I'm gonna lecture recombinant DNA today. And then I think, although we also had like other techniques, this will be, I think, the last topic for the exam. So it'll be proteins, recombinant DNA, transcription, translation, DNA replication. That's a lot of information. Gene expression, it's a lot. So the study guide has those first areas, but I need to kind of add on these last lectures. I just wasn't sure exactly where we were going to get to, but after today I know exactly this is what we covered. So this will be the last topic, but you're still responsible for proteins and enzymes. Go back and look at that. And I will even kind of start maybe a discussion thread so you can ask me questions over the long weekend. Send me an email. Send it to my K Rosales because that updates on my phone. If you send it to Canvas, sometimes I don't always log into Canvas every single day. So make sure just email me directly, and I'm usually pretty good at responding if you have any questions. So, and also as far as DNA, I'm going to remove some of the stuff from the study guide because it was on the last test. Really what you need to know about DNA structure is if I give you a DNA sequence, can you give me the complementary DNA sequence that's supposed to go with it on the other strand? That's really all you need to know for that. And then the percentage of bases. Remember we did it on the board, like, if you have 60% cytosine, how many thymine do you have? Those are the tail end of that lecture. Okay.
So DNA replication, would that be on the test?
Yes, okay. Yes. So DNA replication, DNA structure, we had already covered it pretty, in a lot of detail in macromolecules. So like I said, just the application side of it, of DNA structure. So the application of DNA structure, DNA replication was the next topic that we discussed. Then we did transcription, translation, those two processes. Then we talked about gene expression. And then today, and then the proteins and enzymes lecture. Oh my gosh, this is a lot.
Yeah.
And then finally, recombinant cloning today.
But I know you're going to cut some of the questions off because you're down, right?
Yeah, I'll just have to condense it. So there can't be a ton of questions on every topic. It'll be exactly like the last time. I think that was a perfect amount. It was about 30 to 35 multiple choice and some short answer, 10. So I might make more multiple choice and maybe fewer short answer for this one, just so that we can have, cover everything, but make sure that it doesn't run over time. Okay, so that will be that. I will update the study guide probably this evening with everything up to this lecture. And then anything that we start Monday will be the next exam, exam 3. I tend to feel like exam 3 I think exam 2 is packed with the most information. Exam 3 is a little bit more, these are the types of biotech, these are the ethical considerations. It's a little bit different because we've talked about all the details. Now we're just talking about how those details are used in biotech. So I just think this one will be, you need to really just make sure that you know replication from transcription and translation and gene expression, because on a test, students start mixing those things up. So really make sure, go online, go to Quizlet, do practice multiple choice questions, and make sure that you're recalling the right info, because it all starts to merge together when you see it all in one place on an exam, okay? So make sure you do practice questions and really get that going. Do the study guide for 5 extra credit points. Even if you did well on the first exam, don't just be like, oh yeah, I have this. Do it, because you never know. And then at the end of the class, people are always emailing me in a panic, like, Dr. Rosales, I'm at an 88. Is there any way I can get an A? And you earned what you earned, right? And if you didn't do any study guides, and maybe you forgot to do study guide 2, that would have given you those extra points. So that's where it is. Like I give more than enough extra credit built in that there's really no like, yeah.
I noticed on the study guide, you have something like be able to, show the complementary basic.
Exactly.
Do you want us to draw that out?
You don't need to draw it out. I'm just reminding you, we did examples of that in class. So that's what I'm expecting. You need to be able to apply it. You can write a couple examples out for yourself. You don't need to include anything there, but you should practice doing that. I put it there so you practice. Okay, go back to the lecture, do it again, go back to your notes, practice the examples, okay? All right, so let's do molecular cloning. I put up an updated lecture online and I shortened it, and so I just tried to condense it so we can get through it. And so there's an updated version that's now on campus, and I'm going to lecture that, okay? And so we already talked a little, if you're in lab, you already heard this spiel, yesterday for like an hour. If you're not in lab, then this is the first time you're hearing this, okay? So that's one of the advantages of taking them both at the same time is you hear everything almost twice, but hopefully it really sinks in. And so molecular cloning is a process that is going to involve many different steps, okay? And so, oops, I don't know why I put that there, so I'm gonna start here because I don't know why I put that there when I was messing with it yesterday. Okay, so cloning. I mentioned this in lab, but if you're thinking about a clone, what's a clone? We already talked about this, but refresh. It's a copy. It's a copy of something, an identical copy, right? The Clone Wars, Star Wars fans, right? The stormtroopers, they all look like little clones, right? So what we're talking about is, in the big picture, it could mean an identical molecule, an identical cell, or an identical organism. Right? We've, in some of my other classes, we cloned a plant, right? We took an African violet and we cloned it using tissue culture methods. And we made a whole bunch of the same plant from a leaf. We just cut a piece of its leaf and we put in the right conditions with growth factors and we grew clones of those plants, okay? So you can clone an organism if you want to. What we're really talking about today is the molecular level. We're talking about making DNA, right? Identical DNA molecules. That's what we're talking about here. And I mean, if you really think about it, this has been going on for a really long time. How often do you make clones of plants? You have a plant that you really like. I do this in my own garden. I cut off little pieces of my succulents and I just start growing them or stick them in the ground other places, and there you go. I just cloned my plant, right? You have a whole bunch of them. So, like my brother-in-law gave me a bunch of succulents. I kept them, and then I started planting them all around, and so I have all his clones, right? And so that's been going on forever, right? Making copies of an original plant. What we're talking about is we're talking about cloning at the molecular level. We're making copies of DNA, okay? You're gonna make cells, you're gonna make copies of DNA. And so this is important because it's kind of encompassing DNA replication, so the idea of making copies, but then we're also incorporating the fact that if you have the DNA there in a cell and you have the instructions, can you then make a protein? You can, right? And so two things can come out of molecular cloning. The first is that maybe you just want a whole bunch of a piece of DNA. The easiest way to do that is to put that piece of DNA into a bacteria and let the bacteria do the work, right? And then extract the DNA out from it that you want. The second thing that you could do is once that DNA is in there, the bacteria already has the instructions. So that bacteria can now undergo transcription and translation, and you could even make that useful protein, extract it out, sell it to somebody, or study it, okay? So we're gonna go through this process in a lot of detail right now, step by step. So molecular cloning, okay? So molecular cloning, that's what happened. That slide's out of place. This was supposed to be separate. This was supposed to be the second slide. I'll move it over. Okay, so the process of molecular cloning is here. There's many different steps. There's about 5 steps that you're gonna go through. And I'm gonna walk you through each of these steps in detail, okay? And so let's look at the overview and then let's go through each step by step so you understand what's happening. So step 1 is you're gonna work with something called a plasmid. Okay, what was a plasmid? You guys, we heard about plasmids yesterday in lecture. What are plasmids?
It's a bacterial DNA.
They're bacterial DNA. Are they linear or circular? They're circular pieces of DNA, and are they large or small? They're small. Are they part of the bacteria's genome? No, they're extra, right? They're extra. And so remember that scientists have extracted useful plasmids out of bacteria so that we can do molecular cloning, okay? And so here's a little plasmid. It's a double-stranded piece of DNA. And so really the idea of molecular cloning is that you're going to cut this circular piece of DNA open, and you're going to insert a gene from somewhere else. This looks just like what you're doing in the lab, right? You're cutting one plasmid open, and then you're taking a piece from a different plasmid and you're sticking it in. The other thing you could do is, you know, if you want to study like human insulin, you could extract genomic DNA from human cells, and then you need to find that area on the DNA that has that human insulin gene, and you need to take it out, and then you could glue it in right here. So it's a lot of copying and pasting. If you want to study a gene, then the best way to do that is to get your gene, isolate it, stick it in a plasmid so you can make a bunch of copies of it, and then if you really want to study its functionality, then you can make protein and study what it does. Okay? That's why you do this. You're trying to study a gene, you're trying to study a function, or you're trying to make a whole bunch of protein or DNA. So here's the first step. You have your plasmid. You're going to have to cut them open with little molecular scissors, and you're going to have to stick this new gene in, because you're trying to build a new recombinant plasmid with this gene that you want to study or make the protein from. And so green, so you cut and cut, you're gonna cut and cut. Just remember, big picture is that those little overhangs need to be compatible. Remember we talked about that yesterday? They need to match up. It's like puzzle pieces. Our puzzle pieces, can you just stick any two things together, even if they look somewhat the same? I mean, if you really think about puzzle pieces, they do look very similar, right? But there's very, unique features that make them fit together well. Same thing happens here, and we'll review that. So you cut, and then you paste that gene of interest in, okay? So that's the second thing. You're gonna cut and paste, that's this step here. Once you make what we call a recombinant DNA molecule, because now you've recombined DNA from two different sources. The purple was DNA from a bacteria. That's where you got the plasmid from. Well, this green, that could have been from a different bacteria. That gene could have been isolated from a sea anemone, like we're doing in lab. It could have been isolated from a human. Who knows, right? Maybe it was already being held in a plasmid for safekeeping and stability. And so this is your recombinant molecule because you've recombined DNA from two different sources, okay? Once you've made your recombinant molecule, you do this. You insert this back into a bacteria. You have to get it back inside. We're going to talk about how. You get it back in. Once it's inside, then you let the plasmid, well, first you have to determine whether it got inside. That's step number 3. So you get it in. How are you going to know if you got it in there? I'm going to tell you how, right? Can you look at a flask of bacteria and be like, oh, these look great. It must have all gotten in. I know, there's no way of knowing that, right? You can't tell just by looking at it. And so you will need a way to select for those bacteria that took up the DNA. It's not a perfectly efficient process. Not every single bacteria in that flask will take up your recombinant, so you need a way of selecting. We're going to talk about how you do that. So we use an antibiotic to kill off, and I'll tell you how, to kill off the bacteria that didn't get your plasmid, and only those that did will survive. Okay? And then once you know it's in there and you've selected, then you can do different things at this point, right? Now you've inserted your recombinant DNA, and so you can use the bacteria as your little factory. Right? And that's what this is. You can grow a bunch of this bacteria. Remember you guys plated your bacteria in the lab and you got those little colonies? And that took how many days to even just get like a nice sizable colony? It took a little while. But what you do is you pick that little colony off of that plate and you dip it into a liquid broth, okay, with the algae nutrients, the antibiotic, and you grow up, a liter of it. And then you can even scale it up more, right? You can go further. You can then take that liter and inoculate 10 more liters and grow it up. And then now you can harvest your protein from the bacteria, meaning you'll break those bacteria open and you purify out your protein. Or if you wanted DNA, you break those bacteria open and you purify out this plasmid DNA that you just stuck in. There's different protocols for what you want to do. Okay? So this is the big picture, right? This is the big picture of how you clone a gene. I want you to see it so that you see the big picture before I start giving you all these details that don't make any sense to you. Okay? So any questions about just the general flow? This is the big picture idea. I'm going to now go through each step and tell you about the details. Okay? So let's start here with cut open the plasmid and paste in a gene. Okay, that's the first step. And so, sorry about that. I put that out of place. So here's a picture of a bacteria. And remember that these are your plasmids. They're small circular pieces of double-stranded DNA. They are separate from the genome, meaning all the DNA that the bacteria needs, right, to have all the instructions. It's separate. So not every bacteria has the same plasmids inside of it, the same number of plasmids. They're all different, right? And so these are kind of almost like accessories, that they're extra, right? The bacteria can live without those plasmids, but some of them have more of them than others. Researchers realized that these plasmids exist in bacteria, and they were like, hey, let's use these, right? Let's use these as a tool in the lab. Because they're small, they're easy to manipulate, and you can cut them open, and you can stick stuff inside of them. And so not all plasmids are useful for molecular cloning, right? You can't just go to any bacteria and be like, oh, we'll use that plasmid. Because the plasmids have to have certain features. Remember we were talking about that in lab, that there's certain things that need to be on that plasmid to make them really, really useful for working in a lab. And so the plasmids that we work with have certain characteristics. And remember that these plasmids are capable of replicating by the same mechanisms that we talked about as far as DNA replication. Okay, so once they're there, they can make copies of themselves, and they can also undergo transcription and translation to turn genes on. Okay?
They also can do what?
Exactly. They're in there, so they have access to all the machinery. They have access to the polymerases and the helicases and the RNA polymerase. They have access to everything. So they're just much easier to manipulate. This is kind of a big jumble of genes, right? This is a nice small little unit that you can easily look at, okay?
Are all the plasmids in there the same, or would they be like?
They can be different. Yeah, so remember I was telling you that we used to study bacteria that we isolated from aquatic environments. And so yeah, every bacteria had different plasmids inside. That's what was making them resistant to antibiotics in nature. So keep in mind that bacteria have been around for a very long time, and they need mechanisms to survive. And so sometimes they will acquire plasmids, and those plasmids often have antibiotic resistance genes on them. So if a bacteria is living in an environment where maybe there's an antibiotic being secreted by something else, maybe a different bacteria or some other thing in its environment, it's protecting itself. It's finding a way to survive. And so yeah, some of the bacteria that we looked at had multiple different plasmids that coded for different resistance mechanisms to these antibiotics. And so we had to actually go through the molecular analysis to figure out, yeah, what plasmids were there, what genes were on those plasmids that were actually giving them resistance to ampicillin, penicillin, streptomycin, a whole slew of antibiotics. So they can be different, okay? They can be. And so these plasmids were extracted from bacteria, but we've actually kind of stepped in and intervened and made them even more useful. We've engineered them a little bit to make them more suited for what we need. And so plasmids, it says, can be engineered to be more useful. And so oftentimes you're gonna hear the word plasmid and vector used like interchangeably. And I do it also. Sometimes I'll just say plasmid. Some people will say vector, and you might be like, what? Why? What's the difference? Okay, really a vector is just a plasmid that's been engineered. right? They've put something in it, they've added something to it, it's been engineered in some way to make it more, easy to use or useful for scientists. Something to kind of think about is that all vectors are plasmids, but not all plasmids are vectors, right? That's a good way to think about it. They're all double-stranded circular pieces of DNA. The vector started as a plasmid, but we just engineered it to have something extra or useful. Okay, so they are all plasmids, but a vector has just been manipulated in some way. Okay, so most vectors, if we're going to use them, usually have been manipulated in some way to make them easy to use. And the most common vectors that you can go online, just so you know, there's a site called Addgene. You can go online and you can search through thousands and thousands of vectors that scientists use across the world. And if they engineer a plasmid and turn it into a vector with something really useful, they can deposit their plasmid there. And anybody in the world can go online and look at the maps and see what they've done and buy it for about $60. And they send you the bacteria and you grow it up and you extract the plasmid out, or the vector out of it, and use it. So that's how we get a lot of our vectors that we use in the lab. Okay, so there are 3 very general common features that you'd want your vector to have if you were going to use it for molecular cloning. 1, origin of replication. You guys remember that? What does the ORI do? Remember when we saw the ORI? What happens at the ORI?
There's a bubble.
There's a little bubble, right? The DNA bubbles out and it allows the recruitment of the different enzymes that are required for DNA replication, for making a copy of the DNA. So if you don't have an ORI, is it going to be easy to replicate that plasmid or that vector? No, you need an ORI so you can make copies of it. So you want your vector to have an ORI, okay? You also want what's called, and I mentioned this yesterday, a multiple cloning site, or MCS, okay? This is a short stretch of DNA that has many sites for the restriction enzymes. The restriction enzymes are the scissors, okay? So if you had a vector and it looked like this, maybe, I'm going to just kind of show you, like maybe in this area, maybe you have an area where you maybe have like a BAM-H1 site, or a HIND3 site, or maybe an ECO-R1 site, or a SAP1, or a ECO-RB, and they're all in a very close area. This section would be called the MSC, multiple cloning site, because that means that you could cut, you have so many options where you could cut and paste, right? Because you're going to have to find a strategy that works so that you can cut with the same enzymes here and cut your gene with the compatible enzymes and stick them in. So a multiple cloning site gives you that capability. In addition, most of the enzyme cut sites that are found in the multiple cloning sites are not found elsewhere on the backbone of the vector. Why would that be important? If you're going to cut and insert something here, why is it important that it's not found anywhere else? So it doesn't cut some other place, because then you had a circle. If you cut it twice, now you have 2 separate pieces, and so it's going to be hard to paste stuff in. Ideally, you just want to cut, once, or if you're going to cut twice, they need to be in close proximity so that you're only losing a very tiny piece, and it can open, and then you can insert stuff in. Does that make sense? So this is, again, something that's engineered for ease of use, so that if you do cut with BAM-H1, it's not going to be found anywhere else and cut anywhere else. Okay? So that's the multiple cloning site. The ORI, which you also need, okay? So you'll have your ORI. Okay? And then something that's also very, very important is a selectable marker. It's going to be another gene on that plasmid. And the selectable marker will help you differentiate which bacteria have taken up your recombinant. Once it's all built and you put it back in, that selectable marker is going to help you. Okay? It's going to help you differentiate. Most of the time, we use an antibiotic resistance gene as the selectable marker. And I'll talk about this a little bit more. And there's a picture on the next slide. Okay. There's a lot of other things that we talked about in lab that I'm not including on here right now. okay? This is a very, like, the basics that you need in a vector. What are some other things that we talked about that would be extremely useful, especially for gene expression? Because some vectors are good for just making copies, okay? They're more of like a replication vector, like you just want to make copies of it. This would be suitable for making copies of DNA. Would this maybe be the most ideal for inducing gene expression? What am I missing if we wanted to induce gene expression, have an expression vector?
Regulatory gene, the promoter.
A regulatory gene if you wanted to regulate gene expression, and also the promoter so that RNA polymerase could be recruited to the right site and turn the gene on. Okay, so that's kind of what I'm showing here. This right here, this is like the basics. You need this like at the most primitive. And this vector would be good for replication, for making copies of DNA that are held in here. But if you wanted to express a gene, DNA, RNA, the protein, okay, DNA, the RNA, the protein, you need a little bit more. Does that make sense? You need a few other things in there. So look at this vector. This has a promoter. What does the promoter do? Nobody wants to do the dance, right? It's going to bring the RNA polymerase in so that you can do transcription, right? And then so if you insert the gene here, that's perfect. Your promoter is right upstream, and then there's your inserted gene. And between the promoter and the gene, there's probably going to be an operator in there so that you can regulate gene expression. Does that make sense? Good. So you have that. Here's your ORI, right? Here's your origin of replication. Your multiple cloning site is here. It doesn't show you, all the enzymes, but it says restriction site, restriction site. So this area in here, there was probably multiple restriction sites that you could have cloned into. So that would be your multiple cloning site. This is a selectable marker, your antibiotic resistance gene. Remember yesterday we talked about kanamycin resistance gene in one of our plasmids and an ampicillin resistance gene in the other plasmid that we were working with. Some vectors, if they're really fancy, can even have a secondary selectable marker, meaning like a color change. And so there could be other things that could be engineered in. Oftentimes this will be over here in the multiple cloning site. So for example, if you clone the gene in, then the color will change because you've disrupted some type of gene that was there. So they kind of put this in a different spot just for making it simple. But you could have other selectable markers on your plasmid to make it even more obvious that you got what you needed. Okay, so these are the basics. You need an ORI, you need a multiple cloning site, and you need an antibiotic resistance gene, or more generally, a selectable marker. If you do want to make a protein though, if you want to express, then you also need a promoter. You'll need that promoter, right? So you're going to need that promoter in front of your gene of interest, and you might even need the regulatory gene if you want to have an inducible or repressible. So you'll have a regulatory gene. What was our regulatory gene yesterday in lab? Does anybody remember?
Like one?
In lab, remember, it was called ERA-C. Remember, it was ERA-C, it was upstream, and we haven't really talked about it. So really, I just want you to know this, like the basics, but for lab, remember that if you want to make a protein, you need a few other things, right? You would have an expression vector that makes sure you have your promoter, everything's going on, and you can actually induce gene expression. Okay, so that's the plasmid itself and what you need. So now let's talk about cutting open your plasmid and inserting your gene. So here's your plasmid, and so it's a circle. And if you're going to cut it right here at this restriction site, then now it opens up. You guys see that? So it was a circle. You cut it once right there, and look, it opened up. Okay? Here maybe is the gene that you extracted from a human. Let's say that's your insulin gene that you're trying to clone in. And so notice that you also have here, you have to cut it so that the little overhangs are compatible with the plasmid, okay, or the vector that you're putting into. And so you have to cut the plasmid, you have to cut the gene, and you have to cut with compatible enzymes so that the pieces fit together properly. Okay, that's the cutting part. Then you need to actually paste it in. Does anybody remember what pastes when we do DNA replication? Ligase, right? It's the same thing. The ligase is the glue. Remember, the ligase is the glue. So what you do is you'll have to mix those two things together in a tube, throw in a little bit of ligase in there, and it'll glue that piece in, right? It'll glue it in. So now you've made what you call a recombinant plasmid. You had your original plasmid and you've added in a gene from some other place. Right? Here it was human insulin gene. Okay? So let's talk about the details. So how do we cut? To do that first step of cutting, you need something called restriction enzymes. Remember, they're the scissors. They're the scissors that are going to cut, and they cut at very specific DNA sequences. Remember, we went over that yesterday. They find a very specific sequence, and if the whole sequence is there, then they'll cut at a very specific spot within the sequence. Okay? Remember that, and I don't know if I mentioned this, but just it's really interesting. You know where we get restriction enzymes from? Our good friend bacteria. So we get the plasmids from them, and then we also we can get restriction enzymes out from them. Okay? So restriction enzymes are found in bacteria. They are a defense mechanism for bacteria. So those restriction enzymes protect bacteria from infection from something called a bacteriophage, a virus that can infect them. So if you're getting infected by a virus, usually you're going to die if you don't have some type of mechanism if you're a bacteria, because the virus really just takes over. And so what happens is, if this bacteriophage comes and tries to infect that bacteria and it injects its viral DNA into the bacteria can find that DNA and it can use those restriction enzymes to cut up the virus's DNA. Okay, that's cool, right? I mean,
Don't eat me in.
right, yeah, like, you know what, you're gonna try to inject your viral DNA in here? Like, I'm gonna fight. They don't have a sophisticated immune system like we do, but I mean, they need a mechanism to survive, right? They do, so that was their defense, restriction enzymes that cut DNA. So, I mean, I'm gonna show you in a slide or two, you might be thinking, well then why doesn't it chop up the bacteria's DNA? If those enzymes find DNA sequences and cut, then wouldn't they just cut up everything and kill the bacteria in the process? The answer is no, bacteria are smart. Too smart, which is great. Most restriction enzymes cut at palindromic sequences, meaning that, let me just, hold on. Palindromes, like for example, GAATTC, well, it reads the same forward as it does backwards, because what would be the palindrome for that? GAATTC, that's going this way, 5' to 3'. Okay, if we do the complementary on the other strand, what is it? CTTAAG. So if you read in this direction, is it the exact same sequence? Yes, same forward as it is in reverse. So they're palindromic sequences. Okay. And many restriction enzymes can produce what we call sticky ends, but they can also produce blunt ends. And I'm going to show you a picture of that. So sticky ends would mean that once you cut, like let's say you cut here and you cut here, okay? You made a cut right there. If you pull those pieces apart, this is going to be one side and this is going to be that other side, right? You pull them apart. These are sticky ends. They're those little arms. Remember I was showing you guys yesterday, little arms that stick out. Those are sticky ends. Sticky because are they going to be able to kind of grab on and interact a little more easily? Absolutely. Okay, blunt ends, what does blunt mean? It's just a straight cut, right? You get a blunt cut of hair. Your hair is just like straight across. Okay, so there will be no little sticky overhang. It'll just be a straight cut. Those are harder to stick together because there's nothing kind of grabbing on, right? So that's important. So here's showing you why restriction enzymes don't cut the bacteria's DNA, but they do cut the virus's DNA. How smart is that? That's awesome, right? And so what happens is that, let's say you have the scissors, and here they are, and they recognize this sequence, GAATTC. Okay, and they always cut between the first G and the A. So they're cutting here on this strand, and then GAATTC on the reverse complement strand, you cut there. And so if you do that, right, then it looks like this. You've just cut it open. Okay, this would be the virus's DNA that you're chopping up, but the bacteria is like, nah, I'm protecting myself. That's why I'm even, using these restriction enzymes. So what they do is they actually add on some methyl groups. So a methylase will methylate the DNA, so it'll add this group onto the DNA. So now you imagine you have the DNA. If that enzyme tries to sit down and clamp onto the DNA, there's this big bulky methyl group that's there. So can the restriction enzyme clamp on? No. Okay, so in short, bacteria protect their DNA because it's methylated.
Yeah.
Yeah, and so when that same enzyme tries to go and cut the bacteria's DNA, it's protected. Okay, so that's how it differentiates between the virus's DNA and the bacteria's DNA. Important. If you ever take 191 with Dr. D, he's 100% going to ask you about DNA methylation. He always does, so I'm bringing it up just so you're kind of aware. Here's a little picture of just what it means to have sticky ends or blunt ends. So let's say you are going to cut this piece of DNA with EcoR1. So you're going to look for GAATTC, and you're always going to cut between the first G and the A. Yes? So you read along, you read along, you read along. Oh, here we go, GAATTC. Yes, is that the sequence? And then you're going to cut right here between the first G and the A on that strand. Then you have to go to the reverse strand, GAATTC, and you're going to cut right there. And then remember, this whole section is one unit, because what's holding these two pieces together? Hydrogen bonds, right? So that's going to keep it together, and so you'll pull it apart, and notice you just made what kind of ends here? Are these sticky or are these blunt? Sticky, right? One hangs off a little bit more than the other. Those are sticky ends. This is an example of a blunt cutter. Small one, it recognizes CCC, GGG, and then it always cuts between the middle CG. So if you look at that, channel your inner restriction enzyme, right? You read along, read along, read along, CCC, GGG, ah yes, I found you. And on the other strand, CCC, GGG, ah yes, I found you. And you always cut between the middle CG, or it cuts, and we're going to often. But there's thousands of enzymes. And guess who sells them? A biotech company. Remember I was telling you yesterday? That's a biotech company's whole business. They purify out restriction enzymes and they sell them to people. Okay? New England Biosciences, my favorite restriction enzyme site. Right? You can go on there and search, thousands of enzymes. And so they are named, because you might be thinking like BAMH1, HIM3, EcoR1, where is she getting these weird names from? They're abbreviations, so they are named for the microorganism which the enzyme is produced. So here, Bacillus, this is called then BAMH1, okay? Colostridum, E. coli, EcoR1. This is a different E. coli strain, so it has EcoR1, this one's EcoR2. Okay, so on and so forth. So that's why they're named this, because they're named for the bacteria that they were isolated from. Okay? And then they each have their very specific recognition sequence and the location in which they cut. Does that make sense? You never have to remember this. I will always give you what the enzyme recognizes, where it cuts. You just need to be able to cut and tell me the outcome of the cuts. Okay? Okay, so that was the cutting part. What happens next? You just cut the plasmid open, and you cut your gene so it has compatible ends. What do you do next? You glue, right? You paste. You have to paste it in. So ligase is the glue. It's the glue that's going to paste that in. And so just imagine this is your gene of interest in blue. This is your insulin gene that we talked about on that first slide. And look, it has this sticky end here. This is the plasmid sticky end on this side. And you're trying to glue those two things together. And it's important to notice, do the sticky ends, are they compatible? Do base pairing rules still work? Well, let's find out. If you push them together, this C pairs up with, well, let's merge them, there it is. This first C pairs up with that G, yes? Because this is the sticky end and the sticky end. Did it all match up? Yes, C to G, T to A to T, C to G, yes? And what pulls them together is hydrogen bonding, right? But then you throw the ligase in the tube. And the ligase is this little purple molecule, and what kind of bond is it going to repair right there? Covalent. You remember that from DNA structure? The ligase is actually adding on that covalent phosphodiester bond that's on the backbone. Does that make sense? So I talked to you about it. So when you look at this, you're like, ah, yes, the chemistry. It's building that bond. So you have one continuous piece of DNA. It all matters, right? The chemistry matters. Can we do the molecular biology without the chemistry? No, there would be no molecular biology without the chemistry. Okay, and so that's why you need it. That's why I taught you those things, so this stuff makes sense to you. You understand it broadly and in detail. You can put it all together. So now, that's why there's this dark black line there. You just built that covalent, you just formed that covalent bond. That's what the ligase did, and that's what the ligase did, because remember, that's where there was a little bond missing. When you push those two pieces together, you needed to merge them together, glue them by forming that phosphodiester bond. Okay, good. We good there? So restriction enzymes are the scissors. Ligase is the glue. Okay, what's the next step? We cut and we pasted.
Put it back in
Now what do we do? Put it in who?
the bacteria.
You gotta get it back into your little bacteria, your workhorse, your very low budget workhorse. Yes. It's important, right? You gotta think like, all right, how can I make a bunch of it? So yeah, we did this part. Woohoo! Okay, now we have to insert the plasmid into a bacteria. Any ideas? How are we gonna do this? What do you have to get across if you're a plasmid or a recombinant piece of DNA? How do you get into the cell? Can you just freely float in? Is the membrane gonna allow you to just like, hey, let me in, I'm here? No. So how are we gonna get it in? It's an important question. You need a way to get it in. Someone said heat shock. Good, yes. Heat shock's one option. Did anybody else read the lecture, the readings about how else you could get it in?
Add calcium to the,
So the calcium is important with the heat shock, and then she also said electricity or an electric shock. Yes, those are both ways, so we'll talk about that. Okay, so again, we cut and we pasted, we made our recombinant DNA molecule. Now we're trying to get that's what our DNA stands for, it's your recombinant, you're gonna get it back into your bacteria, okay? This is where we're at. So this is what you're doing. You did the ligation, and that glued the pieces together. And so what you do is once you've glued those pieces together, you're going to mix those glued pieces with your bacteria. Okay, I'm going to tell you about these bacteria because you can't just use any bacteria. They have to be what's called competent. Makes sense, right? What does the word competent mean? Be capable. They have to be capable of doing this. Okay, so you can't just like, oh yeah, I'm going to take this bacteria and mix it with DNA, our DNA, and boom. No, it has to be able to do it. Okay? It has to be competent if you're going to do that. And so the most common method that people use is a calcium chloride treatment, which I'll show you, and a heat shock. And it's really simple. You guys are going to do that next week when you come back for break, right? You did the cutting yesterday. On Monday, you'll do the pasting, and then on Tuesday, you'll do the let's put it back into my bacteria. So you'll do all of these steps next week. You'll finish this whole process out if you're in the lab. And so the goal is to get these little plasmids back into the bacteria. And so if you look at this, did all of them take in your recombinant? Of these 4 bacteria, did they all take in the plasmid? No. How many of them took in the plasmid? Just one, just this one, okay? Not everybody takes in the DNA, okay? Not all of them do, just some of them do, okay? So this process of getting that DNA, that recombinant DNA into this bacteria is what we call transformation. You're transforming it. You're getting something in, okay? And so, again, not all bacteria want to take in DNA from their environment, or they're not all capable. In order for them to do this, they have to be competent. We call them competent. This sometimes occurs naturally in just out in nature, but you know, in the lab, do we want to leave this up to chance? No, right? We want to make sure that our bacteria are competent, very competent. And so you can either make your bacteria competent yourself, or you can buy competent bacteria from a biotech company. They'll sell you competent bacteria, and they are expensive. So in grad school, I was in charge of making competent cells for the lab. So I'd have to make a batch, I'd have to screen them, I'd have to confirm that they could efficiently take up recombinant DNA, and then that would be the batch that we used for the next year to year and a half. I would spend like, you know, days making competent cells for the lab. Okay, and if they were not that very good, I'd have to start again. And I would usually save them for experiments that were relatively easier to do and didn't need as high of a level of transformation efficiency. But yeah, I've had to make competent cells many, many times for my lab. Because if you buy a little box of like 10 tubes of competent cells, they are hundreds of dollars for like 10 little tubes of competent cells. If you make them, it costs you a fraction and you can make a lot, okay? So the way that you make cells competent is you have to treat them with a chemical. So if you want to make them competent, you can treat them with a chemical. Really, you need calcium. And what we usually do is a calcium chloride solution. And so you add calcium to these cells. So you pretty much grow these bacteria up, and then you treat them with calcium, and then you freeze them. And you put them in a very cold freezer, and then you just take them out when you need them. If you are talking about, you know, how this all works, well, the first step is making them competent by adding the calcium chloride to them, right? And so you can do that, and then you keep them and you freeze them, and then when you're ready to use them, you take them out, you mix the DNA and the competent bacteria together, and you do what's called a heat shock. Okay, so this step right here. So you take your plasmid, your recombinant DNA, you mix it with your competent bacteria, and you do a heat shock. What do you think that means? What is a heat shock? All you do is you warm them for like 45 seconds. That's it. You go hold them in like a warm water bath, or you put them in that little heat block that we used yesterday, 42 degrees Celsius. You just warm them up for 45 seconds, okay? And in that moment, those 45 seconds, what happens is the heat and the calcium on the outside of those cells will cause changes in the plasma membrane. And so they don't know exactly the very, very detailed mechanism of how it works, but they think that that calcium kind of opens little pores in the membrane, and for just that 45 seconds, the recombinant DNA can kind of float in, right? It makes like a little pore in the membrane, so recombinant DNA can flow in for just that time. So that's how it works. Yeah, it's a short time frame. The minute you take them out and put them back on ice, the plasma membrane goes back to normal, okay? So that's what's happening there. Okay, the other method, which is not as commonly used because you need a very specific machine, is called electroporation. But what you're doing in that situation is the same thing. You have your tube, you have your bacteria, and then you have your recombinant DNA in there. And what you do is you stick it in this little machine called an electroporator, and you push a button, and it administers a very quick electrical shock to your cells. And that electrical shock also causes changes in the membrane for just that one split millisecond that allows the DNA to just float in. It's really hard on the cells. A lot of the cells die because you just shock them for like a millisecond, and they're not happy. They really aren't. But you can get DNA in. Again, this makes small little holes in the membrane, and our DNA can float in, but you need a specific machine. So that's why that's not as commonly used. Most people do the heat shock method. That's commonly used. It's pretty easy to do. Okay, any questions? That's what transformation is.
Shouldn't that say Cl2?
It should, but it could be, you could treat with calcium chloride, calcium phosphate. You really just need the calcium portion of it. Yeah, so it just depends. If you look up protocols, oh my gosh, various calcium solutions, other things that you mix in. I mean, we were going to make competent cells, and I probably came across like 10 protocols that were slightly different but do the same thing. So yeah, as long as you have the calcium ions that can actually kind of coat the membrane, you're okay. Okay? But yeah, thank you for pointing that out. Okay, so this is just a little schematic of everything that I just told you. And so here you have your bacteria that are competent. You see your competent little bacteria. They've already been treated with the calcium chloride solution. Okay, so they're competent. You see those little holes? They have the potential. Okay? The calcium chloride upon heat shock should make their membrane porous or have little holes. So you have your competent cells. You throw in your recombinant DNA into that same tube, and then what do?
Warm it up.
Then you warm it up. You give them a heat shock. So you literally, we have a water bath, you just hold your tubes in there for 45 seconds. You'll do it on Tuesday, and then you put them back on ice. And during that 45 seconds that they're at 42 degrees Celsius, see the rDNA was on the outside of the cell, but after the heat shock, it goes in the little pores. And hopefully, some of the bacteria have taken up your recombinant DNA.
So why is it only 45 seconds if it goes over?
It just depends on the competent cells, but yes, they've done experiments to show, like for the competent cells that we use, we've shown that more than 45 seconds actually decreases the transformation efficiency. If you don't do it long enough, decreases transformation efficiency. So there's kind of an optimal time frame. And not every set of competent cells is 45 seconds. I've used some that are a minute, I've used others that are 30. Just depends on those cells, and if you've optimized it, you know what that time frame is. But yeah, it can decrease the efficiency.
So one bacteria can take up multiple plasmids?
They potentially can take up multiple plasmids, because if they have multiple pores, you know, depending on how many plasmids get in, that's okay, because then they just make more copies and they have more to start from, right? So it's okay, they can have more than one plasmid. Yeah, there's no way to ensure just one gets in, right? It just, none may get in, one may get in, 5 may get in. It's just, you know, it's mixed in a tube and whatever interacts, okay? And so now, after that cold heat shock, then those, if you're lucky, you have your recombinant DNA inside your bacteria. Okay, good? So we got it in. So now, once we get it in, you have to select for the bacteria that got it, right? The ones that got it. And so remember, you're gonna do this because of that selectable marker. Your antibiotic, you'll want something. And the easiest thing to use is an antibiotic resistance gene on your vector. Okay, because if you have a bacteria, if you have a big bacteria right here, if you add some ampicillin onto that bacteria, is it going to survive that antibiotic? No, right? It shouldn't. It should die. Okay, because ampicillin will specifically kill that bacteria. But what happens if now, during your transformation, you got your little recombinant plasmid in, and now it has that selectable marker, that gene that confers ampicillin resistance. The R stands for resistance. What happens if something's resistant?
It won't die.
It'll survive. It won't die. So now if we dump a whole bunch of amp on this guy, on this bacteria, is it gonna live or is it gonna die? It's gonna live. It's gonna live. What happens if we threw on kanamycin, a different antibiotic, by a mistake? Let's say we grabbed the wrong tube and we threw in kanamycin by mistake. What's going to happen now? They'll die. The resistance is specific to that resistance gene that's in your vector. Does that make sense? So that's how you select. That's how you select. So if you go back to this picture, if we selected here, and let's just say that that resistance gene is in red, and let's say it's ampicillin, which of these is going to die if we treated these bacteria with ampicillin? Is this going to die? Yes. Is this one going to die? Yes. Is this one going to die? Yes. Is this one going to die? No, because it has the plasmid that has the gene on it. Okay? So that's how you'll know if your recombinant DNA was taken up, because you kill off all the others, and only the ones that live are the ones that took up your recombinant DNA. So you're enriching for those ones that got it, and you're just killing off everybody else.
And that way you have the ones that do have it.
Right, because what if only 10% of them took up your recombinant DNA? Now you have a bunch of bacteria that didn't, and so that's not going to be great. You know, you're only going to make 10%. You might as well kill all those off and know that you have 100% of them with what your product and grow those up. okay? So it makes sense, and it's really quick, like you can select overnight. You just put them in the media with the antibiotic, and by the next day, only the ones that live will grow out, okay? And you guys will do that on your Petri dishes. You'll do this exact same thing, and after the transformation, you'll plate them on your Petri dishes, and the Petri dishes will have the correct antibiotic in them, and it'll kill everything that didn't get your recombinant DNA, and it'll only keep, just like this, it'll only keep, only the colonies that grow are the bacteria that had your recombinant DNA in it. Does that make sense? And a colony, what is a colony? It's a clone. It's a little bundle of clones, because really what happens is one bacteria, as you're spreading, right, one bacteria is there, that bacteria turns into two go into four, you know, it divides exponentially, and a day or two later, you start to see the visible presence of that white little colony, because one bacterial cell probably divided into millions that are now just stacked up there forming a colony. Does that make sense?
Okay.
And it's resistant to the antibiotic because of the gene that you put into the vector. Yes, so there's its gene. So only these bacteria that took up your recombinant plasmid with your resistance gene will grow. Yeah? All right, we're almost there. Woohoo! Okay, I've never done this lecture. This is a new one, so my timing, I'm hoping I can do it. All right, so the last steps. I kind of lumped these together because I don't think I need to really go so hard into the details. Is now that you've selected and you know for certain those bacteria have what you wanted, well, now it's time to make a bunch of it. Okay, now it's time to scale it up a little bit. So what you do is from that little plate, you pick one of those little colonies and you can dip it in a liquid solution with the nutrients and antibiotics. So I can kind of draw that out for you. So it's much, if you want to grow bacteria and you want large quantities, the best thing to do is grow it as a liquid. Okay, as it doesn't have to be on a petri dish. It can grow in a liquid. Bacteria are more than happy to grow in a liquid. So what you do is you have your little petri dish and you have your little colonies, right? And so then you're going to get a little tube and you will add your media broth in there. And so remember we used Luria broth or Al-B. So Al-B, this is just a nutrient broth that has everything that the bacteria need to survive. It has salts, sugars, everything, right? It's the food. Okay, this is the food, but it's in liquid form. Okay, and then you pick this little colony like on a little loop, right? You pick one, and then you bring your loop over here, and you dip it inside of there, and you drop that little bacteria in. So now that bacteria got in here, and it'll start replicating. And so you shake it overnight. The other thing you need to add in is the antibiotic. So you just keep that selective pressure on. So you also still need to add in the antibiotic. You add the antibiotic in here as well. Oh, sorry, I should draw it there. So you add the antibiotic in.
And you do this in the autoclave?
So you do this using sterile technique. Okay, so we have a Bunsen burner, right? You open that plate, you use your clamshell method like we practiced when we streaked out our environmental culture. So you have your Bunsen burner on, a flame, you make a little sterile area, you open your plate. Yes, the liquid media would have been autoclaved. It would be completely sterile media that's never been opened so that nothing can grow in there. And you look at it and check it. If it looks murky, well, it's contaminated. If it looks nice and clear and it's been autoclaved, you're good. Okay, but you open that tube with your sterile environment, you pick your colony, you dip it in, you add the antibiotic in, and then you close the lid, and then you turn the flame off and you go shake it. You'll usually put it in a little shaker so that you can get oxygen and it's all moving around and those bacteria are getting everything that they need. Does that make sense? Okay, so that's exactly what this is showing you right here, is you're going to take one of those little colonies and you're going to move it into that liquid culture with antibiotic because you want to keep that selective pressure. If these were grown in Lb plus amp, what should be in this flask? Lb plus amp. Okay, you need to keep that selective pressure going. If this was grown on Lb plus kanamycin, what should be in this flask? Lb plus kanamycin. The thing that makes this plate solid is you add in something called agar. You add the agar in that makes it solid. okay? If you just do LB, it'll be liquid. What makes it solid is agar. So these are LB agar plates with antibiotic. This is just the LB nutrient broth plus the antibiotic. Does that make sense? Okay, and then you grow up a big flask, maybe you grow up a liter of them, and at this point, there's a couple of different paths you can take. If your goal is to purify proteins, well, at this step, you might want to induce gene expression. What does that mean to you guys? If you're inducing gene expression, what do? Are you turning it on or are you turning it off?
Turning it on.
You're turning it on. You need to induce it, okay? So what is that? How are you going to turn it on? We learned about operon, so if this is in an inducible operon, let's just say for example, and you want to turn this gene on, what do you need to add into that flask? An activator, some type of metabolite that can inhibit the repressor, right, and turn gene expression on. There's lots of different, it depends on your vector, what that is, right? It depends what you're working with. So yeah, you could add in something to induce gene expression. Not all vectors are inducible or repressible. Some of them are just constitutive. They're just on, okay? But like some of them are. The one you're going to use in the lab is inducible, and we will add in a specific metabolite called arabinose. It's a sugar, and that'll induce gene expression. Okay, so you'll want to turn gene expression on or induce it, and that's what this is showing you. Oh, look, there's the mRNA, which then is translated into your protein, and your bacteria was just a little factory for you. It did all the work, right? And so now this whole flask has been induced, and you've made a whole bunch of proteins in there, in the bacteria. How do you get the proteins out? Any ideas? How do you get them out of the bacteria? They're a cell. They don't just spill out.
Filter it.
Maybe filter it, but that still doesn't release them.
Another.
How do you get the proteins out?
Break open those cells.
You have to break them open. You have to disrupt the cell membrane and break them open so that you can then purify the proteins out, okay? So you guys will also do that step. We call it lysing, breaking open the bacteria and releasing the proteins and then doing some other steps to purify them, okay? The second choice, if you grow up a bunch of bacteria that have your recombinant DNA, is maybe you just want to make a bunch more of that recombinant plasmid DNA, and you don't want proteins. So the other option could be you grow this all up, but instead of inducing gene expression, you don't induce gene expression, and you just extract out the DNA. How do you think I got all the plasmid that you guys are using in your lab? I had to do that whole protocol, get here, but when I got here, I broke the bacteria open and used a kit to purify out just the plasmids. And I didn't induce gene expression. Okay, does that make sense? All right, how much time do I have? Am I almost there? Yeah, no, I don't have any time. Okay, good thing we were at the end. So you want to just look at these last two slides. All this is showing you is, I just wanted you to look at this one. It's just scaling up. This is just understanding that biotech companies do this on such a large scale. They go through exactly what we just did. They make that 1 liter bottle, and then they'll use that bacteria to infect 6 more bottles, and then maybe a huge 5 liter bottle, and then they'll take that all the way into bioreactors that can hold up to 3,000 to 10,000 liters of cells that have that recombinant DNA in them. They can then break them open, purify proteins, and sell them to people. Got it? Or the DNA and sell it to people. Yeah? All right, you guys. Have a great 4th of July. Be safe.